This is episode 84 of the Immunology Podcast, T Cells and Inflammation in Cancer with Dr. Jeffrey Rathmell. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Jeffrey Rathmell from Vanderbilt University on the podcast to talk about his research on immunometabolism in inflammatory diseases and cancer. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
STEMCELL Technologies would like to introduce you to Human Immunology News. It covers everything from immunotherapy, autoimmunity, and adaptive and innate immunity. Human Immunology News keeps readers current with the latest news, research, policy, events, and jobs related to the immunology community. Subscribe for free at www.humanimmunologynews.com. Happy birthday. It's too soon, right?
it is. It's soon. Yes, it'll be right when this airs or thereabouts. Yes. Now I'm just, in my 40s.
Wait, are you not going to be in your 40s anymore?
I'm still in I'm just like, turning 40 was one thing. Now it's just the slide of ever decreasing numbers in the 40s, which is,
one of the stories I'm going to share today might be very relevant to your situation. I hope that this will help you.
If it can reduce arthritis, I'm all game.
Well, you never know. Maybe. But any plans, any celebrations?
I don't know. My wife's in charge of my birthday plans, and I don't know what's happening. It probably won't be the level of surprise birthday I did for her a few months ago. But, we'll see. We'll see what happens.
Oh, that's very cute. That's just organizing something for your birthday, you know?
So we'll see what happens.
Yeah, well. Why don't we enjoy one of the last days of your...
Being 40.
Of being 40. With some science.
All right.
You want to start?
Sure. So I had to call back to this paper last time on the show, where I was like, hey, there's this really cool paper that just came out that we don't have time to read. But now I had time to read it. And so I'm going to talk about a humanized mouse that mounts mature, class-switched, hypermutated, and neutralizing antibody responses in Nature Immunology.
No way.
Published the 25th of June. First author is Daniel P. Chupp, and last author is Paolo Casali. This is, the newest generation of humanized mouse, but it's pretty dang cool. So there isn't actually a huge amount I need to talk through here. It's really the highlights, but the highlights really tell you why it's awesome. So they used a grafted non-gamma radiated, genetically myeloablated KIT W-41J mutants, immunodeficient pups, with human cord blood CD34 positive cells. So that's the first bit. So this KIT W-41J mouse is a known mouse that has immunodeficiency that they've used for transplants before, but no one's ever really gone all in to really make this work. They've done RAG knockouts as a background for humanized mice. This mouse is a much better ablated system that you can ablate and go.
So, but you still need to. You don't need to irradiate them. Isn't that
you chemically ablate them and then?
Okay.
So that's nice, right? And then human cord blood, but then they follow it with a 17-estradiol conditioning to promote immune cell differentiation. So use estrogen, and these mice work. And so the entire paper talks about how it reconstitutes a human lymphoid and myeloid system. You got marginal B cells and germinal center B cells and follicular helper T cells and neutrophils. And there's lymph nodes and intestinal lymphoid tissue, including Peyer's patches, which look different in humans and mice, and human thymic epithelial cells. They have diverse B cell and T cell antigen receptors.
All human.
Uh-huh.
Quite impressive.
Mature T cell dependent and T cell independent antibody responses. They have somatic hypermutation and class switching. There's some differences between what you see in humans and regular and these mice in terms of the propensity of different IgE immunoglobulin classes. But yeah, plasma cell differentiation and B cell differentiation, and then they challenge these mice. You get a neutralizing antibody responses to salmonella with a flagellin vaccination. So if you hit them with flagellin and then give them salmonella, they respond. If you give them the human Pfizer COVID vaccine and then give them COVID, they're protected. They have the normal human secretion of human cytokines at physiological levels. So they also can develop human lupus autoimmunity with a pristane injection that behaves very much like human lupus does. So the whole point is that the estrogen supports human immune cell differentiation and maturation of the antibody responses, and that these mice are then going to be a platform for using humanized mice. Oh, and their microbiome shifts to look much more like a human microbiome.
God, is there anything this mouse cannot do?
Mount a murine immune response.
I mean, I think just for our listeners, and I don't know if the paper talks a little bit about because there are already humanized or quote unquote humanized mouse models out there. So my lab has a mouse model, but this mouse model does not have lymph nodes. So that is a problem for us.
It probably doesn't have plasma cells running around that are
I don't think so. We're actually working on a grant to see if we can elicit antibody responses. But now I'm thinking, we should.
There's a mouse.
Just tell our listeners why are you so excited? What are the limitations of previous?
They were all half-baked fundamentally, right? So you would either have some residual murine component left, you'd only have part of a transfer, inducible, this myeloablation here is more robust in some ways than just hitting with radiation and trying to kill everything because it's genetically mediated.
Yeah.
And so
and also because for those that won't work, so you have to irradiate the mice when they're like just born or something insane, and it's super difficult to catch them at the right time. So you have to irradiate them, and you need to reconstitute them in crazy. The technicians working with our mice hate working with these mice because it's so specific and the times are so tiny that if you miss your pups and they're too old and they grew over the weekend, it's too late. I don't know how it's the case for this mice.
Yep. So it's an interesting, neat way of doing it fundamentally, and it works. So it's just, and the other thing is they have the immune repertoire to respond, right? So when you see the microbiome shift to be a human microbiome, that tells you something, right?
Yeah.
Tells you the immune system's driving the microbiome environment and it's mapping over. You get responses to a vaccine. So you give exactly like what you'd expect. You get the normal immune responses you'd see in a human.
Yeah.
Including memory. And that's just not what we have to date in any of our models.
Yeah. That's very exciting.
It's super cool.
Where is this mouse being developed? In which place?
The University of Texas Long School of Medicine in San Antonio, Texas.
Well, guys, grab your boots. If you want some good mice, go to Texas. All right. With that, why don't we move to the next talk? I was going to say to the next paper, because as I mentioned, I think you're going to find it very interesting, particularly for you in your advanced age. Not easy being growing up. You cannot be as young as I am. I understand, but there might be a hope for you in the horizon. And it is described in this paper published in Nature, plain Nature, 11th of June. Inhibition of IL-11 signaling extends mammalian health span and lifespan. And the first authors are Anisa Bidjaya and Weiwen Lim, from corresponding author Stuart Cook from the Singapore Medical School in Singapore. And sometimes, and I think, and also Duke. So Stuart Cook is at Duke University in the US. I don't know. This is one of those stories that you say, wow, this is like too good to be true. So let's run through it. Um, as I mentioned, age, one of the hallmarks of age is basically inflammation, particularly, chronic, what is known as chronic sterile inflammation. And it's a central hallmark of aging. And we know, I think by now, dear listeners, I think I already mentioned this many times. Inflammation in old age is not good. It makes it makes your body tired. It makes things that makes you old. And so, in the anti-aging whirlpool of news lately, there's a lot about people, they take rapamycin, they take metformin, these things that activate certain signaling pathways that are associated with blocking inflammation, um, particularly pathways just, like such as mTOR1 activation, and then in a more immunological sense, JAK-STAT pathways are also associated with inflammation. And so this work looks at a particular cytokine that I had never really thought about, which is IL-11. And IL-11, if I understand correctly, has some similarities to IL-6, and it is also pro-inflammatory. It is pro-fibrotic, and it has been to some extent associated with age and with kind of age-associated pathologies. And it has been kind of observed to be upregulated in older people and some role in senescence. So they've decided to really look closer into what is IL-11 doing in aged mice. So what they see is that they have, mice, they let them age. And the first thing they look, good figure number one, is they look at IL-11 expression over time in different tissues. For example, liver, white adipose tissue, around the one that is very commonly used for in mice, for particularly in male mice, is gonadal white adipose tissue that is known to be inflamed in later age in older mice, and also in skeletal muscle. So they look at all these tissues and they see that in principle, you see an upregulation of IL-11 in all of these tissues, and activation of certain markers, certain pathways that, as I mentioned, are associated, ERK activation and downstream phosphorylations of RSK, inactivation of AMPK pathway, activation of mTOR pathway. So this is all checks out as they expected, particularly in male mice. And they show that so what they do, they have this IL-11 receptor alpha knockout mice, and they compare how do these mice look in old age. And lo and behold, they have a lot of his hallmarks, a lot of his activation of inflammatory pathways are reduced. So the litter-mate controls, showed this aging that the IL-11 receptor alpha knockout mice did not. So these mice seem to stay kind of younger. They had lower body weights, decreased fat mass. And what I thought was very kind of impressive is that they also looked into telomere lengths and mitochondrial DNA copy, which are kind of molecular markers of aging tissues. And they also see kind of a difference, a preservation of a younger phenotype in a knockout mice. So in principle, they just do all these experiments. They work on, they study. They also show that they can, to some extent, imitate this by treating the mice with an IL-1 receptor alpha antibody, so a neutralizing antibody. And this, to some extent, also reduced the aging. And they had mice that were around 75 weeks old, so kind of middle-aged mice, and they show that treatment for 25 weeks with this antibody actually reversed a bunch of the inflammation and a bunch of the markers of age. They show that they can lose body weight. So once they're already kind of 75 weeks old. So basically, they change the composition. So they show that among the things that it does, it changes composition of the white adipose tissue. So it kind of reduces by have a more oxidative fat. So I think it will be more like kind of this kind of ground fat. So I think that's probably a way by which this fat is lost. And they do a lot of studies, but I think basically the highlights are that they can kind of rewind some of these aging markers, particularly those that have to do with inflammation, activating of pro-inflammatory pathways. They show that they can extend the lifespan of the mice, either by knocking out the IL-11 receptor alpha or by treating them with the antibody, with the neutralizing antibody. And so they say about a 20% increase in lifespan. So I think it's pretty significant. And what I think is interesting is that they also look, one of the things that they look at, one of the last figures is they look at cancer, development of tumors in these mice, and they show that not only they don't develop more tumors, but they actually apparently have less kind of macroscopic tumors found in these aged mice, which is often a source of mortality for the mice. So they can improve this aging without increasing, for example, risk of cancer, which is always kind of a concern. And so I think, I am looking at see what is the patent that they made when they're going to start, testing this in humans. I'm not sure exactly how much we know what IL-11 blocking would do kind of in a human. I think they also do some blocking antibody with fibroblast. And so that's kind of and they reduce senescence secretory phenotypes when they treat them with this antibody. So I think they are most likely, maybe they're going to go try that humanized mouse, see what happens. So I'll let you know if they start any clinical trials, so you can sign up, Jason.
Yeah, especially if it can reduce my arthritis or something. That'd be nice.
I mean, arthritis is inflammation, so I guess maybe.
Maybe. Although I don't have any inflammation in my body because of my immunosuppressants, so who knows?
Oh, that is true. Well,
and I still have arthritis.
That's annoying.
All right. Well, I'm also calling back to one of your papers from last week on this paper that I'm doing now.
Are you?
So we're talking about TLR4 adapters. Mighty 88's coming back.
Yeah. Okay. So what's up?
In Nature Communications, sphingidine recruits TLR4 adapters in macrophages and promotes inflammation in murine models of sepsis and melanoma. Published July 18th. First author and a corresponding author, Marvin Herring. Last author, Guoliang Xiu. And here we go. All right. So protein interactions, right? That's how you get the proteins to the cell membrane when the TLR4 is, activated, right? Then Mighty 88, as you talked about, makes these structures up the membrane, right? And it's all just protein interaction, right? And that's the story that we all heard. Or is it? So this paper talks about something else that's really important, which are lipids. Because guess what? Often, if you're going to be near the membrane, you want some lipid action going on. So they end up doing an immunometabolism screen and looking at BMDMs as their model system. And they create mice that are knockout in serine palmitoyl transferase long chain base subunit 2. It's involved in sphingolipid biosynthesis. And then they show in the end here that Mighty 88 and TIRAP which are adapter proteins, right, require the sphingolipid or sphinginine specifically to locate to the cell membrane. So that's the main story here, right? So they do a screen. they see induction of that big long name gene, SPTLC2, induces an M1-like activation stage. So, more of the downstream of LPS, more of the NF-kappa B driven signaling. And that's the original work that they have established. They then see that if you knock it out, you have decreases in macrophage cell growth and metabolic fitness. Well, that's not good, right? And well, why is that happening? And then they figure out that MIT88's involved, and the localization of MIT88 looks all wacky. If you knock it out, you don't have LPS-induced recruitment of MIT88 to the cell membrane. Well, that's not good, right? And so you don't have so you have decreased, and you have increased NF-kappa B inhibition, since you don't have the adapter protein to cause the signaling cascade. You have decreased growth, so on and so forth, right? So they do all that work. And then they get an interaction, though. And so they do a biotin trap, right? A biotinylated sphingosine, and they are able to pull down MIT88 and TIRAP, but not other proteins. And they did other lipids, and it didn't work. They did other proteins and didn't pull out anything else. So that was effective, and they did fluorescence and showed co-localization. And then they say, hey, if we give these mice sepsis, what happens, with LPS injection? And you don't have the cascade either, so they don't get as sick and so forth, because you don't have an LPS-induced response. And the same goes with intraperitoneal cecal slurry injections. So yes, if you take a cecal slurry and inject it just like LPS, you also have the same effect, right? So this, bacterial-mediated signaling cascades are not working properly if you knock this out, and it's through a lack of MIT88 recruitment, which they can see in the cells, and in the cytokine arrays of the cells. And then they also see then that you have decrease deficient. If you lose it, you also have decreased anti-tumor myeloid cell activity and increased tumor growth in a B16 melanoma model. So the other end of the coin, right? Like, it kills bacteria and gives you sepsis, but it also kills cancer. That's also broken. Okay, but then the other thing they do is they supplement just the lipid back in the absence of the gene, and show that the fluorescent lipid co-localizes with MIT88 right up to the membrane. So what you're saying is that the TLR signal requires this lipid as a cofactor for translocation of MIT88 up to that membrane surface structure that you talked about last time. That's actually important to know in terms of pathways either to blockade or act upon, but knowing that it's not just straight protein, but you have a lipid a lipid cofactor is a key bit of thing that was not known before today, or before this was published.
Well, the puzzle keeps adding pieces. So then what you're saying is this is kind of what is the word, compatible with what I spoke of last week of these structures being formed. So the MYD88 gets kind of activated by the toll-like receptor, and then it kind of migrates back to the cytoplasm to generate these superstructures. And this is compatible with what they see in this paper.
Exactly.
Well, isn't that nice when there is internal cohesion between different publications? All right. So for the last paper of today, I have noticed that lately I am talking a lot about inflammation and age, and how bad inflammation is. Because in the end, of course, inflammation is an immunological process, but it's so much more sometimes, and it has such a wide ranging amount of kind of consequences. But in this paper that I will talk about, the authors they do a lot of Drosophila work. Interesting. I'm not super familiar with Drosophila, but Drosophila also gets inflammation. And I'll talk in a second about how this is relevant to the human or to the mammalian situation. But in this paper, it's called Infection and Chronic Disease Activate a Systemic Brain Muscle Signaling Axis. First authors, so we have a tetrad of authors: Shuo Yang, Mei Zetian, Yulong Dai, and Rong Wang. From main author Aaron Johnson at the Washington University School of Medicine in St. Louis. And I will try to distill and I'll get too long because I had to do some kind of understanding. So basically, we know that inflammation, and particularly like muscle inflammation, there's correlation between inflammation in the central nervous system and muscle inflammation. When you get sick, when you have infections, you feel muscle pain, muscle fatigue. And we know that the muscle can respond to certain cytokines, particularly IL-6. I think it's a very important factor of muscles. Like it generates they can have this. The muscle has receptors for IL-6, particularly. I think one of the most important kind of myokines that we know. But there is IL-6 is actually a there is a equivalent. There is a homologue of IL-6 that is found in Drosophila. And this is there's 3 IL-6 related ligands, and they're called unpaired 1, unpaired 2, and 3. So APD1, UPD1, UPD2, and UPD3. And they activate so they have a receptor called DOME or DOMless. And they also, so the consequence is activation of JAK-STAT pathway. And it also generates, affects the muscles of the Drosophila. So you can measure that, by measure mobility of the Drosophila. So, and apparently this is kind of, in a way, homologous to the IL-6 skeletal muscle relationship. So that's kind of to understand the context. So in this work, the authors are looking for a kind of more direct relationship between this muscle fatigue and muscle pain and infections in the CNS. And I think this is in a way because we observe that many diseases that affect the CNS also give this muscle component, and this is independent of like an infection in the actual muscle. So he said you don't need that in order to have this IL-6 effect. So they start looking into Drosophila. They infect the little Drosophila with E. coli, and they show that indeed there is this activation of they infect the brain of the Drosophila. There's activation of an innate immune response, and this ends up impairing muscle function, but not because the E. coli is infecting the muscle. It's only infecting the brain of the fly. And they show that there is one of the components that get upregulated. So they see an effect in the skeletal muscle, and they relate this to the induction of this UPD3, one of these homologues to IL-6, and the activation of the JAK-STAT pathway in the skeletal muscle, which disrupts mitochondrial function in muscle. And this makes me think about a story that I also think I spoke about sometime, in which Tregs protect muscle. But in this case, the story was that they prevent interferon gamma from being sensed by the muscle. But it's this idea that you can generate, and this had a consequence of mitochondrial and the disruption of mitochondrial function. So this is very important. Muscles need mitochondria a lot.
Didn't we also talk about how so too much inflammation is bad in muscle, bad mitochondria don't get swole, hence you want Tregs. But then didn't we also have something where you want some amount of inflammation in muscle, otherwise you can't build anything up at all?
Yeah, so there's also a story that I covered before that one, in which there was IL-6 receptors in muscle were also part of kind of normal muscle function. I guess there's some level of course, some balance. But too much inflammation is definitely bad for your muscle. But yeah, there is some level of IL-6, and also Tregs. There were Tregs in the muscle, according to this paper that I spoke about some time ago, express IL-6 receptor as well, and they also respond to IL-6. So, but back to the flies. So in this paper they show that it is this upregulation of UPD3 that initiates the signaling from the brain to the muscle and induces the mitochondrial dysfunction in the muscle. So the infection in the brain is kind of having this effect away from the brain in the muscle tissue. And that this is dependent from ROS. So this is a consequence of the infection inducing reactive oxygen species. And this is what activates after some steps, is what induces the expression of UPD3. So basically, your infection in your brain will affect your muscle through this signal molecule. And they also look into, so they show this also. So most of this in flies. They also, and I think what's interesting is that they try to think about other infections that we know are affecting the muscle, or like that makes you very, very tired and sick. And they're looking into COVID actually, and SARS-CoV-2. There's a particular ORF3A which activates the innate immune response. And after some work, they also show that this ORF3A protein from SARS-CoV-2 can as well affect the muscles. So if they infect the flies with that, the brain of the flies, this also results in effects of the muscle and muscle. I want to say pain. I don't know if that's how they call it in Drosophila, but they see this disruption in muscle function. And again, through a ROS-dependent mechanism, basically, if you have inflammation that activates innate immunity in your brain, you generate this signaling, and that goes directly to your muscles, and that affects it. And then they do some mouse work as well. So I'm looking at the mouse results. So they also have expression. They put this ORF3 in the brain of mice, and they express this protein, and they also show that this has this mimics or kind of recapitulates the expression in mice. So they can also cause muscle dysfunction in mice by not really infecting, by expressing this innate immune activator in the brain of them. And I think basically what they say is that in this case is because of IL-6. So also injection IL-6 directly also makes causes muscle dysfunction in mice. So the conclusion that they come is that if you have these infections that will affect your muscle function, you can block like the IL-6 production that somehow seeps from the CNS into your bloodstream to your muscles, can be targeted to reduce this effect.
I just keep going back to what all the weightlifters talk about with the brain muscle axis and tricking the nerves, and that's what builds hypertrophy over time. It's not just the tearing, but like how you do the lifting and generate mental fatigue and then overcome that really matters for getting swole.
Interesting.
So really, I'm learning a lot of papers today to de-age my body and get buff. Is that what you're telling me, Brenda?
Yeah. I mean, what else are you gonna do? Those are the only options you have right now.
Yeah. All right. Well, we're going to be speaking to someone I knew during training with Dr. Jeff Rathmell at Vanderbilt University in a moment. But before we get to that, are you using macrophages for your research? With Immunocolt SF macrophage medium from STEMCELL Technologies, you can differentiate human monocytes into macrophages. The medium and protocols available can be used to differentiate the human monocytes into M1 or M2A macrophages in a 6 or 8 day culture period. Learn more at www.stemcell.com/macrophage medium.
For our second part of the show, for our interview today, we are very happy to welcome Dr. Jeffrey Rathmell. He is Cornelius Vanderbilt Professor of Immunobiology at the Vanderbilt Center for Immunobiology of Vanderbilt University. He is quite familiar with his work. His lab has extensive knowledge and excellent work on immunometabolism and understanding the metabolism, particularly of T cells. And that's a topic that is very close to my interest. So I'm very excited. Always when there's T cells, I'm very excited. So Dr. Rathmell, thank you so much for joining us today for the Immunology Podcast.
Yeah, thanks. Thanks for having me.
It's a Pleasure having you on. All right, Brenda, you're ready to go on immunometabolism. So
let's just give our listeners some background. I think this is really an authority on the subject, and I think it would be so nice to talk a little bit about why do we care what T cells eat, if we're going to be very blunt about it, or what is the role of nutrition and metabolites and what we call metabolism in T cells, and how has that understanding kind of improved in the past few years?
Yeah, well, it's really changed dramatically. I started my lab, I'd go around tell everybody, oh, what T cells use to get glucose is important. And everybody kind of rolled their eyes and said they didn't really care. So it actually took quite a while to convince people that it mattered, but now people do care. And ultimately what it comes down to is nutrients, metabolites. These are sort of the biochemistry of cell biology. Cells can't do anything if they don't have the right metabolites, the right building blocks, and how they get their nutrients and how they change and how that affects gene expression, all these sorts of things. We're learning more and more all the time. It's, metabolites are signaling molecules. It's totally integrated.
So there's many differences, right? A T cell when it's a naive T cell, a T cell when it gets activated, a T cell when it finds itself in challenging environments such as the tumor microenvironment. We know that has different metabolic programs. So maybe we could start talking about in the lifetime of a T cell, what are the kinds of things a T cell needs in order to kind of fulfill the function that it has at that particular time?
Well, there's a couple of ways to think about metabolism just to start with, right? So if you're taking up nutrients, which cells have to do all the time, right, just for homeostasis, just to live, you're either going to use those to burn for fuel, or you're going to be building, or you're going to store the nutrients. And so a resting T cell, they don't have to build. They're not stimulated to grow. So they're just burning fuel. So they take up things that burn efficiently, and they use those kind of nutrients. When T cells get activated, they have to grow and proliferate. So now they have to switch everything to build. So all the anabolic programs, all the metabolic rewiring happens so that they can make glycosylations and hexosamines and nucleotides and everything else. So they have to do all that. T cells are not really notorious for storing energy. Some macrophages can be, and T cells, there's some data that they can accumulate things like glycogen, which would be a storage mechanism. So mostly it's to grow. And then what we've sort of filled that out over the last number of years is that, all the cells are doing it a little bit differently, and those different patterns then affect epigenetic modifications, post-translational modifications, and then that affects the differentiation and the gene expression. So you start to get these again, this link between differentiation and metabolism starts to become more clear.
And I guess I'm going to be practical here, right? Throw on the clinical hat. What do I want to do with this? So I learn these different metabolisms and how they affect behavior, but obviously I know this isn't the case. So I'm going to go the naive answer here. Like, oh, the cells are living. They clearly have fuel. What's that do for me, the patient who wants to know why you're studying what you're doing to help with disease? If the cells are alive, they're alive. So they're doing their thing.
Well, there's a couple of things. One is, as the cells go through these changes, again, they all do it a little bit differently. So sort of the major goal is to be able to take advantage of those differences, to be able to target the functions of some cells. I mean, if you're in cancer, because you have precision oncology, you want to target a mutation that's unique to those cancer cells. When you're talking about metabolism, it's not unique to that cell, but what you find is that different cells are using different metabolic programs at different times. So you do get a little bit of ability to switch. And, we're starting to see some of these targets that are coming out where you hit one metabolic pathway, you're only going to hit one subset of cells and not the other. So then you can actually modify an immune response. And there's definitely targets that are moving up through the works that way. Now, the other side of it and why you might care is we're learning a lot more about sort of endogenous modifiers, like atacanate is a really good example. So this is a natural metabolite that changes immune cell function. It doesn't have any metabolic role. It's just made from the TCA cycle. And it has this signaling property. That's relevant because dimethyl fumarate, which is it's a real drug, right? That basically mimics what a lot of atacanate does. So there's a possibility to take advantage of some of these endogenous metabolites that do change cell function and mimic them pharmacologically.
There's also some understanding of how, let's talk about a very studied example, which is the tumor microenvironment. And obviously, tumors are also very metabolically active. And I think that to some extent, especially when you're looking into, this activation of T cells, there's a lot of similarities between what a T cell needs, what a tumor cell needs. And then therefore, of course, there's going to be competition and also production of certain compounds such as lactate that are known to be problematic for T cells. So when it comes to, for example, understanding how metabolism will affect the performance of a T cell, in a tumor, and where are the spots that we can therapeutically intervene or change that you think have been kind of the most eye-opening in general.
Yeah, I mean, the tumor microenvironment is a pretty well-studied example. Again, we still don't know that much, but we know more about the tumor microenvironment than a lot of other settings. And cancer cells and activated T cells, they do turn on a similar program. It goes back to the old Warburg kind of model.
Yeah.
But most of that's actually done in vitro, or when you do it in vivo, it's sort of a bulk tissue level. You don't really know what cells are what. So we looked at this and used positron emission tomography or PET imaging tracers, and we saw that actually within a tumor, the most metabolically active cells are actually the macrophages. The T cells are down a little ways, and then the cancer cells even down a little bit more. And the cancer cells are actually much better at using amino acids like glutamine and lipids. So in vivo, it's a little bit different than sort of the dogma of the Warburg and in vitro kind of models. But there's potential there to take advantage of that, because what you see is that T cells tend to use more glucose, macrophages tend to use more glucose, tumor cells tend to use more glutamine, right? So we didn't actually see that glucose itself was terribly limiting in most tumors, but again, tumors are very spatially heterogeneous, so there's probably regions where it can be and others. But things like glutamine and other metabolites might be. And one of the really surprising things is when you target the glutamine metabolizing pathways, this can actually promote an immune response, promote an effector response, both in T cells and make a more inflammatory myeloid cell. And the tumor cells tend to be more dependent on these pathways, so they're hit a little bit more. So this is people like John Powell, Ping-Chi Ho. They both worked in this area. We've done a little bit on glutaminase, but they've used other inhibitors and looked at the pathways. And so that's a pathway where if you target glutamine, you might actually be helping the T cell, helping the myeloid cell by pushing it more towards this glycolytic metabolism. It's more inflammatory, and at the same time, knocking out one of the major nutrients for the cancer cells. And we saw that across a number of different cancer models. So that's not just like one off, but that might be kind of a general principle.
That's something that you mentioned that I think as a general discussion, how difficult it is sometimes, especially when you're studying metabolism of cells, to translate what you see in an in vitro setting, with 21% oxygen, 25 grams of glucose per milliliter in those DMEM media. And how I think the latest research has focused more on trying to adapt these conditions to better mirror the actual, inside of your body. And because when I was reading for my PhD, I was reading a lot about glucose, and all that we know about upregulation of glucose transport is like the key part of activation and is fundamental for the acquisition of effector phenotypes in T cells. And then you have also amino acids. You need amino acids for mTOR activation, things like that. So for those who maybe have, stayed with have not been in the latest updates on our understanding of the differences that we learn from in vitro study when we actually start looking into the in vivo study? What is the most surprising kind of discrepancy between what we thought originally in vitro and we now know better or we understand better when we look closer in an in vivo situation?
Yeah, it probably shouldn't be surprising in retrospect, but when you do all the initial work coming back up for the past 20 or so years, and all these assays are done in vitro with these media that were really designed for cells to grow as fast as possible when people are just trying to optimize cell culture conditions going back decades. There's a lot of glucose there. Glucose is an easy fuel, so cells will use glucose and it'll go all the way through the pathways. When you get in vivo, glucose is still around. We don't really think it becomes that limiting, but it's not that necessarily abundant. There's other things there. So cells use other fuels. And so what happens is they still take up a lot of glucose when they get activated, but they don't burn it the same way. They use it to build. It becomes more of a biosynthetic fuel.
Mm-hmm.
So what you find is that T cells actually use quite a wide range of fuels in vivo. There's a reasonable amount of metabolic plasticity. And a lot of this is because there's a big diversity of fuels in vivo that you don't necessarily model very well in vitro.
Mm-hmm.
And some of those can feed straight into the TCA cycle for energy, and then you can conserve your glucose to make a nucleotide. There's those sorts of pathways. Now, the challenge is when you want to go back in vitro to mimic the in vivo situation as close as you can, you have a couple issues. One is you've got to get all the metabolites right that you can, which is a challenge. Particularly since when part of the reason you have such high glucose in these culture media, these traditional culture media, is that cells deplete it, right? So if you say, well, in vivo glucose is say 3 to 4 millimolar in a given tissue, if you set your cell up at 3 to 4 millimolar glucose, the next day it's going to be 1 to 2 millimolar or lower. So maintaining that is hard. So these cultures are really good, but they're extra challenging. And then there's always variables you can't think about or are hard to mimic, the different ion concentrations. And we've actually been working on the effects of temperature, how that affects some of these pathways too. So you can never get it perfect. You can get closer, but you can't really get it perfect.
This may be a tangent. I'm going to ask you said temperature. So I'm just going to ask this. Do you think body temperature is really 37°C anymore?
Oh, are we changing our body temperature?
Well, my understanding is that from the that was done in the early 1900s where there was a lot more endemic disease. And so everyone was a little inflamed, and true human body temperatures like 36.5 or something, but 37 was true if you all had low level TB or other infections all the time when they did the original measurement. I don't know if you've actually seen that things behave somehow magically better, not quite at 37.
Well, all right. So you could get me going on temperature for a long time, we said, but the trick about body temperature is it's circadian, it's hormonal, it's variable. It depends on where you are, where you're sampling. Is it core temperature? So I don't know that you could say we don't have a fixed body temperature. It's roughly 37 on average, but maybe it's 36.5. I don't know. It is changing over time, but some of this actually has to do with obesity, right? So that actually changes, that can shift body temperature. So body temperature is this big variable that is hasn't been considered a whole lot. A little bit here and there, but not in a lot of papers. So again, it just all comes back to the fact that it's hard to model these things. And temperature matters a lot because every enzyme is temperature dependent and molecular complexes and all these sorts of things. So a small change in temperature actually can have a big effect.
And is the obesity effect due to the shift from brown fat to white fat? Or from just more body mass and so you are bigger?
I think it's a combination of body mass bigger, might be harder to dissipate heat. Also, there's this sort of a chronic inflammatory state that might lead to a little bit of heat as well. There's not really a good answer for that.
So there's a lot to be determined yet. That is, So I don't work so much on immunometabolism anymore, but that is kind of always the feeling I had is like, we know it's so complex. There's so many factors. we literally know less than what it's more than what we don't know than when we know in a mind-boggling way sometimes. But I think that we still have your group and other groups working on the field have really tried the best, to design good experiments to understand at least, different parts of the metabolism. I don't know if you want to maybe just talk about recent projects in your group, because I'm going to keep talking about your old stuff, but I know there's newer stuff. I know that you guys have a preprint talking about fatty acid synthesis. You also studied the effect of obesity on PD-1 and macrophages. Would you like to maybe highlight what is the latest things that your lab has worked on that kind of excite you or
Well, we have 3 major areas that the lab focuses in on. And actually, the lab is actually a little bit large at the moment because we merged with my wife's lab not so long ago. So that sort of expanded the lab. But we work a lot on tumor microenvironment, and this includes things like obesity. So we're interested in how obesity is actually the number 2 risk factor for cancers, only behind smoking. Once you get to modifiable behavior associated risk factors, but it's not understood really how or why. And then there's this whole concept that oftentimes obese individuals will respond better to immunotherapy. So they have a greater risk to develop a cancer, but a better response to an immunotherapy. Not better response to every therapy, but immunotherapy is going to stand out. So that this obesity immunotherapy sort of paradox. So we've been interested in that, and that led to a study where we showed that macrophages would induce PD-1, and it basically do the same thing to the macrophages that it does to T cells, and sort of shut them down, make them more suppressive. So you have this more immune suppressed environment because PD-1 is now turning the macrophages and the T cells off. As soon as you come to an anti-PD-1, everything is sort of unleashed, right? So we're interested in how obesity affects these checkpoints, and how does it affect it in the tumor microenvironment in particular, and the crosstalk between adipose and tumors. That's one area. Another area I mentioned is we're interested in just discovering new immunometabolic mechanisms. So do a lot of CRISPR screening there, and we have a story that should be coming out in Science Immunology relatively soon, where we took the inborn errors of metabolism. So these are monogenic human diseases of metabolism, of which there's like 1,400. And I want to say 81 have been cross-listed as inborn errors of immunity, meaning those 1,400 different disorders, only 81 have a clinically defined immune phenotype. And but we thought that must be woefully underrepresented, just because these are rare diseases, they're complicated diseases. Patients don't really get a good immune workup. Anyway, so you just take those 1,400 genes, put them in a CRISPR screen, activate T cells, and you come up with all sorts of things, right? So there's a huge overlap between these immune disorders and the inborn errors of metabolism. And so that's sort of a gold mine of new mechanisms, and it's all really relevant for these patients because they might have some immune component to their disease that people haven't really considered. And then the last area we've been working in actually has to do with temperature and fever. So this is why I know a little bit about fever. We're very interested in how a fever temperature can be, what does that do to immune response? How it's pro-inflammatory, and at the same time induces this a heat response stress, right? So you got to get this dual effect.
So you mentioned your wife's lab, which is a good segue to ask a little bit about that. So in full disclosure for the audience, I happen to have known you and your wife for a while from my time at UNC when your wife was a co-director of the MD PhD program there.
Yeah.
And you came along with her to various retreats and told us youngins, younger than even Brenda is today at the time for me, how to try to have survive a dual career, or scientific or academic career, and a lot of advice therein that I've used in my life quite successfully, since my wife's also a scientist. So as a segue, your wife's an NIH director now, and maybe you could talk more on the recent of of how you manage something, how you guys have been able to manage both your careers at a general level and moving around, but also like this is a newest wrinkle, which is kind of I would describe as extra special and complicated, and has some parallels in the business world, but it is pretty unique. So I don't know if you could provide just your thoughts, not necessarily even advice, but just your thoughts on navigating this, because that's a big one.
Well, as we were going through up again the NCI thing, that's a little different, as you said. As we've been going through this for the years. I mean, I've been very fortunate to have a very understanding wife, and the fact that we're both in the same career path, we kind of, she travels, I travel, she helps me with my grants, I help her with her grants. So that's been our model all the way through. And it's a really, really flexible career path too. So as our kids are grown up, we're able to, take turns and, basically you have to be organized to plan it all out. With Kim now at the NCI, that's an extra wrinkle because now she is most of the time in Bethesda, and I'm here in Nashville at Vanderbilt. So we're commuting back and forth, and sometimes she's down here, and sometimes I'm up there. She's going to be down here come on Sunday and be here on Monday. And then the following next couple of weeks, I go up there. So it actually works pretty well because we talk 2 to 3 times a day by FaceTime. And when we were both here, we both were working a lot, and of course you get lots of opportunities to talk, but you kind of take it a little bit for granted. And so actually, in some ways, it may be less time, but the quality might be better, I guess. I don't know. It's an interesting way to think about it. And we have a few patterns that we've worked out. So like in the morning, so since she's in a different time zone than I am, right? She's East and I'm Central. Rather than just call each other because you don't know what they're doing, we'll do the Wordle, the New York Times Wordle, and you can share your Wordle results. So you share the Wordle, and when you share your Wordle, then you're ready. I'm awake. I can talk now. So I had to be careful because I'll do my Wordle. And so it's also interesting. You have to do a puzzle to talk to your wife. So I do a puzzle, and then if I get it, then I'll send her my Wordle, and usually within 20 or 30 seconds she calls, or vice versa, and then we'll talk for half an hour or so to sort of catch up in the morning, and then usually sometime in the day we are able to talk, but always her schedule is super busy and kind of crazy, so that doesn't happen as often. But then in the evening we'll talk again, after work and then before bed. So actually it's okay.
Yeah.
It's complicated. The logistics are crazy because our kids are older. They're 29 and 24. They both live in different cities. So coordinating around them, one of us is constantly on the move. But it's fun.
Must be, I mean, having the right support is so important, especially with a career such as yours and your wife's. I want to clarify because maybe for our foreign listeners, the NCI is of course the National Cancer Institute.
Okay, yes.
And she's president, and it's also kind of a political appointment. So it has extra layers of complexity, right? Being
it is,
in that chair.
Is the National Cancer Institute director. She's appointed by the president. So Biden appointed her. She has a commission. This is this really fancy document from Biden saying that she is the NCI director. So we have an extra reason to pay attention to the election this year. Now, when Biden's stepping down, and kind of anything can happen, right? But presumably, the NCI is not super political, and we're hoping that she can continue to work there regardless of the election outcomes.
I'll just ask this. Do you find now that there's a political appointee in your household that it changes the perspective or lens or even behaviors that not her, obviously she has a whole set of things that she has to follow, but for you as well as the other half, do you you pause a little more maybe on Twitter, or you really start thinking more about what's going on, or just that the worldview changes a little bit?
Yeah, no, definitely. I want to make sure that we don't, I don't want us to do anything to get her in trouble. We actually have this is sort of informal policy in the lab where I can still apply for NCI grants. I'm actually allowed to do that, but the perception is not good, right? It's not great. So not planning on doing that. I don't intend to
Mm.
do that unless there's some particular reason. So, but what we come down to is because it's legally allowed for me to do it, but it's not a good idea for me to do it. Uh, we tell the lab that they should follow a Tucker Carlson rule, right? It's like, don't worry about if it's legal or not, but just think about what would it look like on Tucker Carlson.
Well,
So,
that's interesting though, because the NCI has so much funding that for like a F fellowship for someone in your lab or something, that can be an interesting extra wrinkle, right? Like you may be able to go to NIA, some of the other institutes. This is getting in the weeds. People know these acronyms. You can go to the Immunology Institute or something else, but the NCI is the giant gorilla of funding sources, and so it's one thing if you're like, well, I'll figure it out, but then for your people, for all these other grants, that can be tough.
well, it hasn't been an issue so far.
Yeah.
We haven't actually applied for an F through the NCI since Kim became the director. And again, it's not, she would recuse herself from anything that would come up from us. And she has to recuse herself from anything from Vanderbilt just because I'm an employee here.
Yeah.
Um,
But
Yeah.
Tucker Carlson rule.
yeah,
like
Yeah.
is the Tucker Carlson. What is it going to look like?
Oh my. All right. I think we're kind of reaching the end of our conversation, but we like to kind of ask a little bit more of a science adjacent question. We already discussed some of that, but I would like to ask you. So, a question we like to ask is, what is the biggest misconception about science that you would like to correct? And I think I might have an idea on what that is.
Oh, you think you know what I'm going to say? Okay, well, I think the misconception is that we tend to work alone, and this is like a single. It's like you go into your lab and you do your work. That's just totally wrong. That might have been the case, decades ago, but now it's a team sport, right? And that's partly because the fields get so complicated. You think, okay, immune metabolism, we have to have biochemistry, we have to have cell biology, we have to have immunology, tumor biology. You got all these fields have to come together, and you just can't do it on your own. And it has to be a group effort. So it's all about team science now. It's not. It's also a lot more fun that way too, because you're working with other groups. And it gets you into the other part that I think is really important is that particularly when you're at a university, I mean, you're a professor, your job is to teach, right? So we do research, but someone asked me just the other day, how would I label myself? And, is it like a scientist, professor? It's definitely professor, right? Because the mentoring, the working with students, that team effort, those interactions, that's what it's kind of really all about. The research is great. That's fun. That sort of happens. Um, but it's really about those interactions between people that it's the most fun.
I thought I knew what you would respond because I have seen some of your tweets about the good things of being
Oh
in
yeah.
science, and of course this is one of the things you did mention. I thought it was very uplifting
Oh,
to
good.
bring that to the table. I don't know if you would like to share some of your favorite, maybe the top 3 things that are good about being a researcher that you think everybody should keep in their mind, especially when the days are hard and then the work is not working.
Yeah, this is not an easy job, right? You know, you get a lot of rejections, and there's other issues that come up. Power dynamics are a big deal, but you don't want to forget the good things. There's always some balance in life is important. We talk about I think the 3 things that are the most sort of thematically important are just the people you get to work with, the team science part of this, really diverse, right? You get people in lots of different fields, lots of different sort of opinions and mindsets and around the world, really. So it's a great way to interact with lots of different people and learn a lot of different things that you could not learn. So that's the first thing. Second thing I think is really important is gets to this idea of teaching. That's really rewarding. I think if you ask any uh, PI who's mentored a number of students or postdocs, once they get sort of a little ways into their career, so they're not focused on not so much focused on are they going to be able to make it per se, but they're going to say that their papers are great, they're happy, they're proud of them, but they're much more proud of their people and the students, postdocs who've come out, and that's the impact. That's the much bigger impact. So that's a real privilege, I think. That's actually one of the biggest privileges being in academia, is you have that as an option. And then the third thing that's great about the job is the flexibility, right? Like I mentioned, when we had our kids and our kind of complicated career paths, the fact that we can kind of adjust things as we want, you have this intellectual freedom to do things as you'd like, to follow these questions that you're interested in. Those are all fantastic. I think that's the lesser of the three, actually. I think that's actually less important than the other two, but at the same time, those are the three themes that I think stand out.
Well said. Thank you. As I said, your career advice on managing with kids is something I deployed.
Yeah, and I think the other key thing about that is you have to have a supportive spouse, but that doesn't just put it on them. That means you have to be supportive too. It's a two-way street. So you have to understand that, things come up. Sometimes people are more busy than others. Sometimes they're more stressed than others, or just are disappointed, and you have to sort of adapt to that. And again, it goes both ways in terms of that.
Everyone takes a turn in all the modes.
Yeah, and it all works out.
Well, on that high note, it all works out, and it's always good to hear, because some days you really have your doubts. Thank you so much, Jeffrey Rathmell from the University of Vanderbilt University, for joining us in the podcast today, talking about T-cell metabolism and career advice. That is gold.
Thank you very much.
Thank you for having me.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunology Podcast or via email at info@immunologypodcast.com. If you have feedback or you would like to suggest a guest, we are open to suggestions. See you next time.